And hello everyone, I'm Runhao from CBU. I'm a second year PhD student. And today I'd like to talk about the new TMS-EEG system at the CBU and how to use it to manipulate our brain rhythms. So as nicely described by Saskia, we have already know that we can always correlate the brain rhythms with specific cognitive processes. For example, people would think that the theta oscillation at the prefrontal cortex is positively correlated with working memory performance. But we don't know whether there is a causal relationship between theta and the working memory. So researchers would like to use TMS, especially the entrainment protocols, which is to use the rhythmic TMS, for example, at theta frequency, and it can entrain the local theta power and phase coherence. So when we compare with the no TMS conditions and with theta entrainment conditions, we may find the differences in their behavioral change in working memory capacity. So we can draw conclusions that, okay, maybe the theta oscillation has a causal role in working memory capacity. But still, we have some concerns about that is how TMS works, because we don't know whether the TMS entrainment can really entrain theta oscillations, and we don't know whether the theta power or the phase coherence can directly influence for example, the working memory capacity. So here comes the TMS-EEG, concurrent TMS-EEG studies. So when participants is doing cognitive tasks, and we can record their EEG signals and have the TMS pulses at the same time. So for example, we can use the theta pulse at their prefrontal cortex and recording the EEG signals. So in this case, we can use directly to use the EEG signals to find the time frequency analysis to see whether the theta power or phase coherence can be changed by theta TMS. And then when we correlate with neural activity and behavioral change, we can then build a causal link from the TMS protocols, neural activity change, and behavioral change. And more importantly, I would say, we can use the EEG data to do some more multivariate analysis, such as MBPA. So compared with the no TMS condition, whether the TMS conditions can decode something better. And then we can consider that, okay, the TMS of the theta frequency can also influence some information coding. Thus, we would say the TMS-EEG systems can establish causal relationship between brain size or neural oscillations and specific cognitive processes and behaviors. And this is just an example of the theta oscillation, so it can do facilitation effects. It can vary around like facilitation and inhibition with different TMS protocols within different tasks. So at the CBU, we've already had a very good standalone TMS system and neural navigation system. And in this year, we get to new TMS-compatible EEG system very excitingly. So to be TMS compatible, we need some extra features of the EEG system. For example, the first one would be the DC coupling recording. It is to avoid any online filtering of the data, because if you do any filtering before removal of the extremely large TMS artifacts, it will largely distort the data. And the second feature is a very high sampling rate, obviously we're about 5,000 Hertz, and by default we use 25,000 Hertz here. That is to prevent the saturation of the EEG electrode, and with the high sampling rate, we can capture TMS pulses and artifacts with a very fast recover, and just less than 10 milliseconds. And this will be very useful for pre-processing steps I will mention later. So here's just a video about our lab, and we can look at it. You can clearly hear the TMS pulses. Yeah, so this is our current studies using alpha TMS to entrain the alpha oscillations in the parietal sulcus. So yeah, we have neural navigation systems always on during the whole experiment, and we have TMS coils and EEG recording at the same time. So as you can imagine, there are many challenges for TMS-EEG studies. And the first one is obviously the acquisition I showed before. And the next one may be how to process the EEG data. And you may imagine that the TMS-EEG will have numerous different artifacts. As shown here, this is just a single pulse TMS. And you can find the very large TMS artifacts, and the remaining will have the muscle artifacts and electrical artifacts and so on. But fortunately, we now already have some standard pipelines to deal with this data. So after pre-processing, we can get normal waves of EEG. So we can use the clean data to do any analysis we mentioned before or you would like to do. So next I will introduce a bit about how to pre-process the TMS-EEG data. So firstly, as I mentioned before, the extremely large TMS artifacts is a major thing for us to remove. So we know that for the typical EEG data, the scale is about 10 microvolts. But for the TMS-EEG, TMS artifacts, it will be about like 10,000 microvolts, extremely large. But fortunately, the large TMS pulses can only last about 5 to 8 milliseconds. So we see good recording parameters. So what we do is just to remove that period of data, because we typically won't use the 10 milliseconds about this data. So we can see that the right panel, we can see when we, after we remove the very large artifacts, we can have some relatively clean data, but still have very large artifacts there. And that one is majorly the muscle artifacts, which shows in the pink curves here. So the next task for us is to remove the muscle artifacts. And what we do for this muscle fact is just do an ICA. And actually, we will do 2 rounds of ICA for TMS-EEG data. And in the first round of ICA, we just remove the TMS-evoked muscle artifacts. And we can have some plots here. So the left one is a single pulse TMS, and the right one is rhythmic TMS with 5 pulses. So actually, the muscle artifacts is very, very strong, and we can clearly find that, for example, it will be very focally at the stimulation point. And for single pulse, it's just one very large shot here. And for rhythmic one, we can have 5 shots here. And when we look at the power spectrum for rhythmic one, we can have the peak value in the harmonies of the stimulation frequency. So we can just use the first round of ICA to remove the muscle artifacts. And then we can have something like reasonable plots of the EEG waves. And then we can do the normal pipelines of the EEG pre-processing. For example, we will first interpolation the data and do pan-pass filtering, and then do the second round of ICA to remove any other eye blinks, eye movement, and electrical artifacts, and so on. So after we get the clean data, we can do any other further analysis from TMS evoked, like potentials and time frequency analysis, and multivariate analysis, and so on. So that's we can see that with standard pipelines to process the EEG data, we can obviously have some good quality of data to do the analysis. So the third challenge, I would say, is to find a good control. As it's always important for the TMS studies, especially also for TMS-EEG. And this is a rhythmic TMS-EEG studies with 5 pulses in alpha frequency. And typically, we will have 2 choices for the control conditions. One is arrhythmic TMS. So in arrhythmic one, we will fix the first pulse and the 5th pulse, and the last pulse, and we only change the 3 pulses in the middle to be arrhythmic. And another choice is to do a sham TMS. So it will be at the same frequency, but it's not the true magnetic stimulation. So previously, the sham coil is always just give you some clicks, the sound of clicks. But actually, participants will always think, okay, that's a fake coil, so I know that. So recently, they have developed a new sham coil, we would say, which calls advanced sham coil, so which is exactly used in CPU, so it's worth for us to look at it. So for the active coil, they have 2 windings, and the direction of the current flow are opposite, so you can stimulate at the center of the coil focally. But for the advanced sham coil here, we also have the direction flows of the magnetic field, but they are at the same direction. So it can avoid the focal stimulation in your brain, but it will more or less stimulate a little bit about the peripheral regions of your brain. So participants can always find, okay, there's still something like magnetic fields there, so similarly, they will have some subjective feelings. So is the sham coil perfect? Unfortunately, actually, no. Previous studies have shown that the sham coil has different physical features compared with active coil. For example, they have different sound. And we can see that if we have the 60% of the output of the active coil sound, it's just equivalent to about 40% of the output of the sham coil. So they don't equivalent in the sound level. And if we would like to do a control condition, it will be worthwhile to control their sound level, right? And so we think it's possible, and it's worthwhile for us to do a replication study or a pilot study to try which one is a good control, at least for these specific coils. And that we did for our pilot. So we just recruit 6 participants in resting state and give them 5 pulses of a TMS in alpha frequency at the right IPS. And we have 4 different conditions. The first one is a rhythmic TMS in alpha frequency, and 3 different control conditions. And one is the arrhythmic TMS, and a sham TMS, and sham TMS with matched sound, which is according to this picture. So firstly, I gave the participants some questionnaires to ask them how annoying this pulse is, and how painful you feel, and whether you feel some twitches. So fortunately, compared with the rhythmic TMS and arrhythmic TMS, the feelings is somewhat comparable. So that's a good news for us that for the subjective feelings, the arrhythmic coil may be the good one to use. And how about the sham coil? Okay, the sham coil, as you can imagine, it will have a little bit stronger feelings, especially for the twitch. So sometimes I can ask the participants, participants will tell me, okay, it's too strong sometimes for the twitch, especially when I use relatively high intensity, like 110 motor threshold. So I would say it's not suggested to use a sham coil at a very high intensity, even if it has the same intensity as the active coil, because it will have stronger feelings for that. And how about the sham coil with matched sound? Actually, it has lower feelings. So it is difficult for us to find a balance, actually. If you would like to control the sound, the subjective feelings will be different. And if you would like to try to find the equivalent point for the subjective feelings, the sound must be different. So it's difficult for you to choose which one to choose, yeah. And next, we will compare the entrainment effect of different ATMS protocols. So when we have the alpha TMS entrainment, so this is time frequency plot in the x-axis, it's time, and the y-axis is the frequency. We can clearly see that each TMS pulse can entrain a broad band of activation, so especially in the high frequency band there. And after the fifth pulse or the fourth pulse, you can find in the alpha frequency, which are around 10 Hertz, there is an entrainment here for the power lasting about hundreds of milliseconds. So yeah, this is what we found. We call it entrainment protocols. And how about the control conditions? So we can briefly think that all three control groups don't entrain such strong alpha power there. And when we subtract using the arrhythmic TMS to subtract all three controls, we can find that there are some differences there. And regarding to the power, so it seems that the sham coil performs better because it has the largest differences here. And we also look at the ITPC results about the phase coherence, the inter-trial phase coherence. And similarly, the alpha TMS can train some alpha ITPC increase there. And compared with all three different controls, we have found some differences here. So at least in power analysis and the ITPC analysis, we can find that compared with the rhythmic TMS, all three controls are good to use. And how to choose which control you want to use may be also depending on your research questions. For example, if I would like to try to examine the specificity of the role of alpha, I will try arrhythmic TMS as a control. And if you just only use a single pulse to perturb something, and okay, you may just have a sham coil. Great, let's have a summary. So concurrent TMS-EEG is a good way for you to use to examine causal relationship between brain size, brain rhythms, and the cognitive processes or behaviors with a high temporal precision. And as we showed, TMS artifacts could be linked with a standard pipeline with proper recording parameters, so don't be too worried about that, and we can do that. And third one, compared with control groups, an active or rhythmic TMS could enhance both power and ITPC, and the choice based on your research questions. So last but not least is that the TMS-EEG system now is ready in CPU for to use. And excitingly, we will have a new TMS Combot here next year. And it can help you automatically with the neural navigation during the experiment. So very, very cool. And yeah, thank you. Especially thanks for my supervisors, collaborators, and the fantastic technical teams. Thank you very much.